Children's Research Hospital. And most of you probably, at least the Americans here, probably know that we are known as a pediatric oncology hospital. But really our newest endeavor in our hospital is really to take on devastating pediatric neurodevelopmental disorders. So I work, so this research I'm going to present is actually not from my group. It's from Dr. Zhuming Peng's group. So he is a member of both structural biology and developmental neurobiology departments in our hospital. Unfortunately, his lab and himself cannot be here today, so they have tasked me with the job of presenting their research. I will do my best and get the message across and answer any question, but I'm happy to give anybody, researchers and families, their information, and they're more than happy to talk with you guys individually and answer questions. So their lab uses mass spectrometry to study the proteome and the metabolome and as well as system biology. So they combine all three together to better understand the mechanism of human disorders. So really, what is a proteome? So this is the central dogma of cell biology or cell science. So our genome, which is encoded by DNA, so that's the building block of life, and DNA is transcribed into RNA, which is what codes for protein, and then RNA is translated to proteins, which are translated to proteins, which they are the enzymes and the proteins that function in our cell. So there are about 20,000 coding genes in the human genome, but on average for a singular cell type, only about 12,000 genes are expressed. So we know about CHAMP1. It is a de novo disorder caused by mutations, caused by heterozygous pathogenic variants that lead to the disorder. And the symptoms that we see in our kids are intellectual disability, developmental delay, seizures, hypotonia, among others. So currently, we know that there's no effective treatments or therapeutic intervention for CHAMP1 that treats the disease at a systematic level. And mostly that's due to our lack of understanding how these pathogenic variants affect the central nervous system or neurodevelopment. So to study the brain is incredibly challenging in humans because we do not have access to brain tissue normally. And if we do manage to get brain tissue, they do not grow in a laboratory setting. So therefore, if we're not able to model something, we're not able to study it, nor do we have a good method to test any potential therapy. So to combat this, what the scientific community have decided to use are these induced pluripotent stem cells, or iPSCs for short. So iPSCs are essentially where we collect a skin biopsy or a blood drawn from patient, and we introduce 4 reprogramming factors or Yamanaka factors and turn them into these iPSCs. So these stem cells really have the potential to generate any tissue of your choice. That could be heart, it could be bone, it could be lung. And in our case, what we're interested in are the central nervous system. So we can make different types of neuronal cells, so neurons, astrocytes, microglia, or together we can make these little mini brains in a dish, which we call brain organoids. So these brain organoids, we can use them as a model where we can track neurodevelopment. We can look to see if any of the disease phenotype recapitulate in our laboratory setting, and we can later on use them to test for potential therapeutic interventions. And this is also great because we know that it comes from this particular patient, so the genetic background in these brain organoids really truly reflect everything about this patient in addition to the disease pathogenic variant. So what the Peng Lab has proposed to do is to collect and generate iPSCs from patients and correct those mutations, so in our case CHAMP1, to have a isogenic control cell line, which is wild type, and then grow them into cerebral organoids, is what they have chosen to do, and then compare the protein expression signature, or the proteomics, between the control as well as the patient-derived organoid, and hoping to find changes in the proteome, and that will delineate any functions that CHAMP1 may be playing a role in neurodevelopment. So they have generated iPSCs from 2 patients. So the first one is a missense mutation that led to a stop codon at arginine 497. And the second one is a 2 base pair deletion that led to a frame shift where a stop codon is introduced as serine 181. And then they use CRISPR editing to correct these mutations to generate a wild type lines as isogenic controls. And the method they've used to generate these cerebral organoids, which a lot of the researchers here are familiar with, is using the stem cell technology cerebral organoid differentiation kit. And they have chosen to grow these organoids all the way to day 40. So at day 40, they collect organoids from these different cell lines. So mutation 1 refers to that missense mutation. Mutation 2 refers to that frame shift that led to a premature stop codon. Wild type 1 is a corresponding control for this one, and the wild type 2 is a corresponding for mutation 2. So this is just a standard PCA analysis to see how different or how close these samples are. And you can see that despite the fact that both mutation leads to heterozygous expression of the wild-type CHAMP1 gene in these 2 cell lines, the 2 lines are still clustered very differently as expected because there are inherent differences between the line, and the rest of the genetic landscapes are different between the line as well. And in terms of wild type, again, they also cluster very differently, which is not very surprising as there are inherent differences between the lines. So the next step they did is to do proteomics profiling to see what is different between the line. So let's just start here. So we have our patient line compared to the corresponding control. They look for genes that are downregulated as well as genes that are upregulated between the lines, and their cutoff was 2 standard deviations away in terms of fold changes, and these are all p-value of 0.05 and below. So as you can see, there's 835 genes that are downregulated and 430 that are upregulated, and same was done for the second set of patient lines. And they started to look to see what's overlapping or what's shared between these differences. They found 157 that are downregulated and 87 that are upregulated. Then they went a step ahead to see to perform Pearson's correlation analysis to see how these change or delta genes are correlate with CHAMP1 expression. So essentially, the Pearson correlation assess how the abundance of the 2 protein changes relative to each other across the samples. A positive correlation means the 2 proteins might have, so with CHAMP1 might have the same, they participate in the same biological pathway or co-regulated. So they generated this heat map, and they found these are the top 30 targets that were considered that has passed this threshold. So they just wanted to highlight some of the interesting target genes. So the first one is catechol O-methyltransferase, or COMT for short. It is a membrane-bound enzyme that breaks down dopamine into inactive metabolites, and its dysfunction has been associated with various neuropsychiatric disorder, including schizophrenia, bipolar disorder, and has also been characterized in terms of abnormal dopamine signaling in the brain. And a second target that they found is HDAC1, which is a histone deacetylase, and it's involved in neurogenesis, neurodifferentiation, and axon regeneration during neurodevelopment as well. And the third targets, or the group of targets they found, are the HNRNP proteins. So there are 3 of them on this list, which is extremely interesting. So these group of proteins has been involved in pre-mRNA processing in the nucleus, and they also affect mRNA metabolism and transport. And various members of HNRP genes has been associated with intellectual developmental disorder as well as other developmental epileptic encephalopathy as well. So these has turned out to be very, this was meant to be a pilot study for the lab, and it turned out to be some very interesting target. And they're currently repeating the experiment with the same set of cell lines. and they want to continue to differentiate the organoids at day 40 to repeat the experiment that they've done, but they also want to continue on to later stages where more mature neuron emerges from these organoids. So they are planning to do the proteomics analysis they've done before to see how robust the data was from the first round, and also identify any potential targets that they have missed. They want to do bulk RNA sequencing to look at how the transcriptome is matching or not matching to the proteome changes that they found. And they're also interested in performing single-cell RNA sequencing to see how the different makeup of the organoids changes, so in terms of different neuronal population, different glial population as well. So this is the Pang lab. The ones in the ladies that are highlighted in the star, they are the ones that performed these experiments and carried out the project. So I'm happy to connect anybody to them to answer any questions and any suggestions that you guys have. And of course, at St. Jude, we're very collaborative. So this is a group effort, or this is an institution-wide effort. So we have our Anjana, who runs the neurostem cell core, so she's going to be taking over in terms of generating more efficient organoids and other type of organoids for the lab. And we also have Chandra Miller, who runs our Center for Advanced Genome Engineering. So what they do is that they correct those patient mutations to generate the control cell line for them to use. So I'm happy to take any questions that you may have. I will try my best to answer. Thank you.
Yeah, that's a very nice presentation. So I think you said at the beginning the iPSC cells, you complement them. You put the CHAMP cDNA back into those cells. Is that your control?
They just corrected the mutation. So one of them is let's go back. Yeah, so one of them is a heterozygous, it's this missense, so they just corrected that missense to get an isogenic control. And same as down below, yeah.
So it's a CRISPR-mediated, some sort of...
Yeah.
Okay, so how do you know they're functionally complemented is sort of the question. You know that you corrected the mutation, but I'm wondering, can you actually go back and look at the expression of the protein now?
Oh, to check to see whether you have... Yeah, they've done that, yeah.
Okay, thank you.
Very, very nice talk. I think one of the comments I would have for them is I think it's really going to be important to do the single cell component of this, because if you're looking in bulk at protein differences, this could be driven by a shift in the proportion of cells. And so knowing that the composition of those organoids is the same will be critical, I think.
Yeah, absolutely. We don't want to take away from the Berto lab, though. I feel like that's what they're doing.
I was just wondering, with the stem cell lines, what is y'all's, I guess, end goal? Like, are y'all hoping eventually there will be, because I know you said there's no treatment now, but will there be in the future for the stem cell lines you are using?
I think the benefit of the stem cell line is that we have a humanized neuronal model to test if whenever there is, say metformin is the one that came out the AI hits, but we need to know what is actually fixing. So the organoids, they do, at least with the other disorders that we work on, for a seizure disorder, they do recapitulate seizures that we see in the kids. So if we found a gene therapy or a drug that can potentially fix the seizure, that kind of give us an opportunity to test. So that's the goal of the stem cell line and generating into these organoids. And really, we with this, because we can track them in a timely manner, so we can see at what stage of development differences start to emerge with the control line.
Also, one more question. I mean, I am a parent with 2 children who have missense, so I definitely would love to, work with you guys and, to, see if we can develop also more research, because I would love to know more about what y'all are doing with it.
Yeah, absolutely. I can definitely put you in touch with Dr. Pang's group.
Okay,
last question, we're running behind.
Can you see morphological and functional differences among different organelles from different patients, like rate of synapses or variation of intracellular calcium concentration or something like that?
So that is the goal, and I think so we can do that with this model, and I think Stefano's lab is gonna tell us more about it. But I think that is what they're planning to do as well, to find to really, because I guess when you find a drug or a therapy, you need some sort of functional readout to test. So it's important for us to identify those robust functional readouts or differences that we can test across different variants and using these organoids.
Thank you.